Hi all. So welcome to another session of Stroke Support India. And we have Professor Bruce here with us again. We have had two sessions with him, great sessions. And we are looking forward to another interesting session today. The topic being a stroke and brain, which is of course a big topic. So we'll be probably having about 40 minutes of a presentation, followed by the chat session that we usually have. And Sridhar is here to manage and moderate the meeting. And I will also be there somewhere in the audience and later on come in. Just a short introduction to Dr. Bruce, just as for the viewers who may see it later. He was a professor of psychology and neuroscience at Lawrence University, as well as a magician, which is a passion he is still also pursuing even now. Suffered a stroke about 10 years ago at age 62, when the doctors did not expect him to live through the night. However, he spent more than a week in coma, followed by intensive care, lengthy nursing stay. But he did return to teaching at Lawrence University for 8 more years before retiring in 2019, and has also followed up on his passion of magic again, and has a mission of raising stroke awareness and inspiring stroke warriors. And we do intend to cover various topics such as brain structure, spinal cord structure, type, post-stroke fatigue, etc. during our meet today. So that will be the broad aspects of the chat that we are going to have. Over to you, Bruce, and Sridhar. Sridhar will be moderating. So to you, Bruce, and you can maybe share your point presentation and take it further from there. Over to you, Bruce and Sridhar.
Okay, well, thank you so much for introducing me. As he said, I did have a stroke about 10 years ago. And actually, on one of the slides, I will show you the type of stroke I had and why the attending neurologist thought I would not live through the night. That might be clear during the presentation. There are many topics that I will talk about, so I will try to keep it under 3 hours in length. No, just kidding. It should be about 40 minutes or so, and then I will be happy to answer any questions you might have. But there is a lot to cover. So I'm going to share my screen and put on the PowerPoint presentation here. Here we go. I do have a PhD in neurobiology. That's why I know something about this stuff. And we will get started. These are the various topics: brain structure, spinal cord structure, neuron structure, stroke type, the impact of stroke location, left neglect after stroke, Gerstmann syndrome, post-stroke fatigue, and stroke recovery. So pay attention because there will be an exam at the end. Okay, Hippocrates, the father of medicine, said long ago in ancient times, suddenly a healthy person is seized with head pain. Immediately the voice fails. He snores, his mouth is open, that is, it gapes. And if someone calls or moves, he only groans, nothing with meaning. He was describing apoplexy, which came to be called stroke around 1800. In the United States each year, more than 795,000 people have a new or recurrent stroke. According to the National Stroke Association, around 2 in every 3 stroke survivors will have some form of disability, and stroke is a leading cause of disability among American adults. Post-stroke depression is the most prevalent psychiatric disorder after a stroke. It affects nearly 1/3 of the survivors during the first 5 years after disease onset. Stroke sufferers are also 2 to 3 times more likely to develop chronic pain than non-stroke sufferers. There is a post-stroke pain syndrome that often affects survivors. It first seemed to be associated with damage to the thalamus, but we now know it can occur after damage to other places in the brain as well. Depressed mood, social isolation, poor subjective well-being, and mental distress contribute to increased motor impairment, disability, and risk of future stroke. Here we have the main divisions of the central nervous system. And as I said, pay attention because there will be an exam. The chart is on the left-hand side. The color-coded brain is on the right. In blue on the right, and in the chart on the left, the top of the brain, what you normally think about of the brain, is the forebrain. It includes the Telencephalon, which is the cerebral hemispheres. Again, that's what people normally think about as the brain. It also includes the Diencephalon in gold. I'll say a bit about the Diencephalon. That includes the thalamus, which is actually where I had my stroke, and the hypothalamus. Then there's the mesencephalon in purple on the right. And then as we get down, going down towards the spinal cord, we have the metencephalon, which includes the cerebellum, which sort of looks like a little head of cauliflower on the back of the brainstem, and the pons. Then there's the medulla or myelencephalon and the spinal cord. Here we have a picture of the left side of the brain. You're actually looking at the cerebral cortex. Cortex means bark. And just like the bark of the tree, the cerebral cortex is on the outside of the brain. In that regard, how can you tell that a tree is a dogwood tree? By its bark. I just said that to keep you awake. Here we go. Now, a cross-section of the human brain shows the inner structure. You can also see the outside. A large groove is called a fissure. A smaller groove is called a sulcus. A bump on the brain is called a gyrus. You can also see the difference between the gray matter and the white matter. The gray matter consists of neuronal cell bodies. The white matter consists of axon pathways. These axon pathways are coated with myelin, and a main component of myelin is cholesterol, which looks white. That will make more sense when I show you an individual neuron in a while. Here we have again an outer view of the left hemisphere of the brain. So the brain is looking forward to the left. At the front of the brain, you have the frontal lobe, again in light blue. In dark blue, the back portion of the frontal lobe is the primary motor cortex. This is the part of the brain that controls your movements. Behind that, in sort of an orange, is the parietal lobe. That's the somatosensory portion of the brain. That deals with touch, pain, cold, and warmth. The front of that, sort of in dark orange, is the primary somatosensory cortex, and this is where you feel things on your skin surface. So if you shake hands with someone, you don't feel their hand until the information gets from your hand up your spinal cord to an area in the primary somatosensory cortex. That's when you feel it. At the back of the brain, the very back, in sort of a purple here, is the visual cortex. That is where you see. So information has to get from the eyes to the primary visual cortex for you to actually see something. And then in green down here is the temporal lobe. That is where your auditory cortex is. That is where you hear something. Now, this describes, in simple terms, a main organizational principle in the nervous system. A lot of axon pathways, beginning or ending in the cerebral cortex, are crossed. That is, areas in the right cortex receive sensory information and control body movements on the left side of the body, and vice versa. This is called contralateral control. For visual information, the left half of your visual field of each eye goes to your right occipital lobe, and the right half of the visual field of each eye goes to the left occipital lobe. That is, if you are looking straight ahead, everything to the left of your center of vision is called your left visual field, and everything to the right of your center of vision is called your right visual field. That is illustrated here. On the left-hand side, you're looking down on top of the brain. On the right-hand side, you have the visual field. And everything to the left of that visual field, sort of in yellow gold there, goes to your right occipital lobe. Well, everything in your right visual field, sort of a gray there, goes to your left occipital lobe. This again is the left side of the brain, the left hemisphere. And this side shows the main portions of the brain involved in speech. So at the front, in the frontal lobe, you have Broca's area. That's the part of the brain, again in your left hemisphere, that controls your ability to talk. When that area is damaged, you have what's called Broca's aphasia or motor aphasia. You have a lot of difficulty talking. Your speech is very telegraphic. That is, just like an early telegraph, when messages were sent via a telegraph, short interconnecting words were left out. And if you have damage to Broca's area, your speech is like that. It's almost impossible for someone with Broca's aphasia to say the phrase, no if, ands, or buts. They can't say those short interconnecting words. You can understand speech, but it's very difficult for you to talk. Down in the temporal lobe, you have Wernicke's area. If that area of the brain is damaged, you have Wernicke's aphasia. In this case, you have difficulty understanding speech. Your speech is very fluent, but meaningless, much like that of a politician. Interconnecting Broca's area with Wernicke's area is an axon pathway called the arcuate fasciculus. It is through that pathway that you talk to yourself and understand it. If that pathway is damaged, you have a condition called conduction aphasia, in which it is very difficult to repeat things that you hear. More towards the back of the brain, sort of in between the occipital lobe and the temporal lobe, is the angular gyrus. Again, this is in the left hemisphere. The angular gyrus converts visual to auditory information and auditory to visual information. If that is damaged, you have a condition called alexia with agraphia. That is inability to read and inability to write. If you're going to read, you have to take a visual image and convert it to an auditory image for you to understand it, because Wernicke's area only understands auditory coding. If you're going to write, you have to take an auditory sound and convert it into a visual one that you can write on a piece of paper. And that again goes through the angular gyrus for that to happen. Is this clear? I hope.
Yes, so far.
Okay. Now this, in very simple terms, shows how the brain divides its work.
Well,
The left hemisphere, shown on the left, that controls your right hand, both your ability to move the right hand and to feel things in the right hand. It also controls speech, language, science, logic, writing. The right hand, on the other hand, controls music appreciation, art appreciation, dance, fantasy, sculpture. It actually also controls the prosody of speech, that is the melody of speech. Our speech has a melody to it or a rhyme to it, and that's controlled by the right hemisphere. Now, the prefrontal cortex is the front of the frontal lobe. It's critical for making decisions, for planning, for evaluating strategies. It's part of your personality, your temperament, your affect. This is the last part of the brain to fully mature. By mature, I mean full myelination. And again, I briefly mentioned myelin. A brain area matures when the axons in that part of the brain develop the myelin coat. In other words, your ability to make good decisions and so on isn't really complete until you're around 30 years old, because that's when the prefrontal cortex is fully myelinated and your personality is intact. This is a midsection of the brain, but I showed this to point out that it is a cut through axons that leave the frontal lobe and go to the rest of the brain. Those axons are severed in a so-called frontal lobotomy. Those were popular in the United States and England in the 1950s. Here's a question for you. What did the alcoholic schizophrenic say to his doctor? I don't know if I would rather have a frontal lobotomy or a bottle in front of me. Okay, next slide. This is the spinal cord. Goes up and down the middle of your cerebral column. It's about the size of your little finger. And you can see the axons going into and coming out of the spinal cord. A cross section of the spinal cord is shown here. The back of your spinal cord is called the dorsal portion, and the front towards your stomach is called the ventral portion. And if you look at the left side of this figure, you see the blue axons coming in are bringing in sensory information into your spinal cord, while the red axons going out are taking out motor information. Now, moving on to the brain itself, the very top of the spinal cord becomes the brainstem. This includes the medulla, the pons, the cerebellum, and the midbrain. This is a half section. It's cut down through the middle. You are looking at the right hemisphere of the brain from the middle portion. And starting here at the bottom, we have the medulla, and then the pons. Stuck on top of the pons is the cerebellum, and then the midbrain. So down here, we have the brainstem. Below the brainstem is the spinal cord. The medulla controls the so-called vital reflexes of the body. It regulates heart rate, digestion, breathing, blood pressure, coughing, vomiting. Drug overdoses often cause death from depression of the breathing center in the medulla. When people die from an opiate like heroin or fentanyl, which is very popular right now, but very lethal, that kills them because they stop breathing. Many people have died from opiates, including Jimi Hendrix, Janis Joplin, and Prince, but many other people as well. It's because they stop breathing when the breathing center is depressed by those opioids. The pons, which is above the medulla, contains sleep centers. Sleep is an active process, and you go to sleep when a sleep center in the pons becomes active. The cerebellum, you recall, is sort of stuck to the back of the pons, and that's involved in the control of movement and balance. It integrates sensory and motor information and controls rapid movements and hand-eye coordination. For example, if you are playing tennis, you have to coordinate the visual input of the tennis ball coming towards you with the motor output of the movement of the racket to hit the tennis ball. And that is occurring through the cerebellum. Now, you're looking at the left hemisphere of the brain, but the point here is the crossed area at the bottom there, going up and down the middle of the brainstem is the reticular formation. That's the part of the brain that keeps you awake. The main cause of coma is destruction of the reticular formation. So a stroke in that part of the brainstem could cause a permanent coma.
Could you point that area, Bruce?
It's called the reticular formation.
And which one is this the dotted one? Which, can you just put your...
It's the striped area there.
Okay, this one, okay. Mm-hmm.
The striped area going up and down the central core of the brainstem. That area is turned off when you sleep, and it's turned on when you awaken. If it's damaged, you don't wake up, because that's the part of the brain that keeps you awake. Okay?
Yeah, sure, please.
Okay, now we move up to the diencephalon. The diencephalon contains the thalamus and the hypothalamus. The hypothalamus is below the thalamus. That's how it got its name. The thalamus receives sensory and motor information and passes it on to the cerebral cortex. So the thalamus is sort of like a relay station. It allows for selective attention. When you're paying attention to something and ignoring everything else, that happens in the thalamus. This shows the thalamus, sort of an egg-shaped structure in the middle of each hemisphere. And this shows how it sends information to the cerebral cortex. The hypothalamus receives information about your internal environment and controls your internal states, what's called homeostasis. So it regulates body temperature, water balance, hunger, thirst, reproduction, and emotional responses. The telencephalon, remember, that's the highest part of the brain. When people think of the brain, they're thinking of the telencephalon. Included in that telencephalon are the basal ganglia, which control slow movements. Remember, the cerebellum controls rapid movements. That'll be on the test, so remember that. And then the limbic system is involved in the control of emotions. Now, this is a section of the brain that sort of goes through the brain like this, and I'm showing you this because it shows the basal ganglia here. Basal ganglia are some structures in the brain. There are actually three: the caudate, putamen, and globus pallidus, that are involved in the control of slow movements. Now, this is a longitudinal section through a rat brain, and I'm showing you this to show you the input to the caudate and putamen of axons. There are cell bodies here in a part of the midbrain called the substantia nigra, and they send their axons to the caudate and putamen. The caudate and putamen are collectively called the striatum. So this is the nigrostriatal dopamine pathway. You have one of these pathways on each side of the brain. The neurons in the nigrostriatal dopamine pathway gradually degenerate over the course of your lifespan. And when 80% of them have degenerated, that's when you develop Parkinson's disease. So if you have a stroke in either the substantia nigra or the caudate putamen, you will get symptoms that resemble Parkinson's disease. Okay? And then there's the limbic system. Again, these control emotions, and there are several of these. I'm going to talk about three of them: the amygdala, the septal area, and the hippocampus. The limbic system was first described by Papes. It looks like it would be pronounced Pepes, but it's pronounced Papes. It's a group of structures that sort of form a ring in each hemisphere. They control emotions, memory, and they're also involved in motivation. And this shows in the left hemisphere, Where the amygdala is, it's at the front of the temporal lobe. The hippocampus is inside the temporal lobe, and the septal area is here along the midline. Now, amygdala versus septal area. If the amygdala is damaged or removed, you have taming or docility. This was first demonstrated in laboratory animals, monkeys specifically. Wild monkeys had their amygdala removed, they became very docile, very calm. Removal of the septal area, on the other hand, causes what's called the septal rage syndrome. I once saw a rat with its septal area removed chase a computer programmer up on a stool. And this guy was about 250 pounds, a big burly guy. He was standing on the top of the stool saying, "Get that rat away from me." The rat was on the floor at the bottom trying to jump up and bite him. So the septal area, if that's damaged, that can cause problems. In the I think it was the 1960s, there was a gunman in a tower at the University of Texas in the United States, and he shot 3 or 4 people with a long-range rifle before being killed. And a post-mortem examination of his brain showed a tumor in his septal area. The hippocampus. This is closely associated with the formation of new conscious memories. What did the hippocampus say in his retirement speech? Thanks for the memories. Do you know who this is? Probably not. This is patient HM. He's the single most famous patient in all of neuroscience. On the left-hand side, we see his brain. This is the underside of his brain, and it shows the hippocampus was removed. On the right-hand side, you see a normal brain, which contains the hippocampus. This is a coronal section, again showing the removal of the hippocampus. This is a normal brain. Patient HM had a severe memory problem. It began in 1953 when the hippocampus was removed to prevent seizures. He had seizures that could not be controlled any other way. So a neurosurgeon removed his hippocampus. And after that, he could not learn any new words, songs, or faces. He could learn motor skills, but he had no memory of learning them. So, for example, he learned how to ride a bicycle after this, but had no memory of having learned how to ride a bicycle, even though he could do it. Okay. Now, this is a picture of some neurons. That's a microelectrode going towards the cell body of one. Your brain has about 100 billion, that's B, billion neurons. Neurons have 3 parts: a soma or cell body, dendrites that receive information, and an axon that sends information away. Okay, this is a picture of a neuron. At the top of the slide, you see the cell body and dendrites which receive information. There's a single axon that sends information away. And this is the myelin sheath surrounding the axon. You could think of an axon as like a 10-foot long hot dog, and each section of myelin would be like a hot dog bun around part of it. Now, at the end of the axon, you have the axon end foot. It's got these little spheres in it. These are called synaptic vesicles. They contain a neurotransmitter. The neurotransmitter is released into a little gap, a synaptic cleft, where it interacts with receptors on the cell body of the receiving neuron. So neurons communicate with each other through a synapse, which has three parts. The presynaptic membrane that contains those neurotransmitter synaptic vesicles, the postsynaptic membrane, which has receptors, and a synaptic cleft, the gap between the pre and postsynaptic sides. And this shows the cell body and dendrites of a single neuron receiving input from other neurons.
Bruce, I will just interrupt for a minute. I think Dhamanji has raised his hand. Let's see.
Oh, yes.
Rajendra Saab, if you have some query, you can put it now, or if possible, we'll wait. But if you want to say something, please do.
He had actually mentioned it's inaudible in the chat.
Right. That we responded to him that it is audible.
Yeah, that's what.
Yes.
Seems to be okay.
I'm not quite sure what the question was.
No, that's what I'm trying to just find out in case he has a query, Bruce. Rajendra Saab, do you have a query? We cannot hear you, sir.
You cannot hear me?
No, I'm talking
No,
to
we're
Rajendra. No, we. So that's okay. We'll carry on. We'll come back to him later, or maybe you can
Okay.
put in the chat, Rajinder ji.
I will
What do you?
continue.
Yes.
A single neuron.
Can you just
single
go back
neuron-
one slide, Bruce, just to maintain the continuity again? Yeah. Can you go back a couple of slides? Yes, that's where we were. Yeah, from here. Thank you.
This one?
Yes, we'll start again from here. Yes.
Okay. This shows an individual neuron. And again, your brain has about 100 billion neurons. This shows the parts. You see the cell body, the dendrites that receive information, and the axon, which sends information to other neurons. And each of these little endings here is an axon end foot. And that releases a chemical, a neurotransmitter, onto another neuron. And that axon end foot has these spheres, these synaptic vesicles that contain the neurotransmitter. You could see a synaptic vesicle rupturing and releasing the neurotransmitter into the synaptic cleft where it interacts with the receptor on the receiving neuron. And again, these are the three portions of the synapse. And this shows a single neuron getting input from several other axons. An individual neuron may have up to 50,000 synapses on it. And it's that collection of synapses that determines the function of the receiving neuron. Now we get to stroke. Reductions in blood flow to the brain of sufficient duration and extent lead to stroke, which results in damage to neuronal networks and the impairment of sensation, movement, and/or cognition. There are two main types of stroke. Ischemic or obstructive stroke is the most common form, accounting for about 87% of strokes. It occurs when the flow of oxygen-rich blood to the brain becomes blocked, primarily due to blood clots. Hemorrhagic stroke accounts for about 13%, arising from leaking or ruptured blood vessels in the brain. This diagram shows the difference. On the left-hand side, you see an obstructive or ischemic stroke. The right-hand side, you have a hemorrhagic stroke. Smaller ones in the left-hand figure, a larger one here, the right-hand side. Now there's several types of brain hemorrhage. First, on the left here is an epidural hematoma. Epidural means on top of the dura, and I'll explain the dura in a little more detail in a minute. But the dura is a tough protective coating that surrounds the brain. So this would be on top of. On the right here, number 2, is a subdural hematoma. That is below the dura. Number 3, lower left-hand side here, is a subarachnoid hemorrhage. The arachnoid is the second layer of the meninges, these membranes that surround the brain. And 4, in here, is an intracerebral hemorrhage. That is the type of stroke that I had, an intracerebral hemorrhage. And that's why the neurologist told my wife I would probably not be alive in the morning. As the blood expands here, it can push the brain against the inside of the skull and kill all the neurons here that are being pressed there. Luckily for me, a neurosurgeon drilled a hole in my skull and put a tube in to drain this blood out. So when my wife came to see me in the hospital, I was on a ventilator and there was a tube coming out of my skull leading to a bag that was filling with blood. Now, getting back to the meninges are layers of tissue that cover and protect the brain and spinal cord. The outermost is called the dura mater. It's a tough protective coating. Beneath that is the arachnoid, which has a space beneath it filled with cerebrospinal fluid. And finally, the innermost one is called the pia mater. It's sort of like a thin layer of saran wrap. It completely covers your brain. And this shows the outermost dura mater. Then beneath that is the arachnoid and subarachnoid space that's got blood vessels. And then finally at the bottom, the pia mater. Okay? The penumbra. The penumbra is an area of tissue that borders the stroke core in an ischemic stroke. During an ischemic stroke, there's reduced blood flow to this area, but neurons there typically do not die. They're damaged, but not dead. They can recover their function when blood flow is restored. On the left-hand side, in the darker area, you see the ischemic core. That's got the neurons that are dead, and the larger area surrounding is the penumbra. When blood flow is restored, if you look at the right-hand side, the central core is still dead, but neurons in the penumbra can function again. And that is responsible for stroke recovery. By the way, with an hemorrhagic stroke, there is no penumbra. What causes a stroke? A common cause is atherosclerosis, or a so-called hardening of the arteries. There's plaque made up of fat, cholesterol, calcium, and other things that build up inside the arteries, leaving less room for the blood to flow. A clot may lodge there. atherosclerosis makes it easier for a clot to form. Hemorrhagic strokes often result from uncontrolled high blood pressure that causes a weakened artery to burst. That is what the atherosclerosis inside the artery looks like. It narrows the area that blood can flow through and can be easily clogged. Just like real estate, location, location, location is important in a stroke. Exactly how a stroke affects a person is depending on what side of the brain it occurs and the amount of damage. Some individuals may experience temporary arm or leg weakness, while others may lose the ability to speak or walk. A stroke in the left hemisphere is more likely to disturb language production or comprehension, which reduces the ability to communicate. So aphasia from stroke generally occurs when the left hemisphere is damaged. When the right hemisphere is affected, the intuitive thinking, reasoning, and problem-solving abilities, as well as perception, judgment, and spatial functions can be impaired. Right hemisphere strokes can make it difficult to locate objects, walk up or downstairs, or get dressed. I had a right hemisphere stroke, and my wife was impressed that I can dress myself in the morning. On the other hand, she was impressed that I could dress myself in the morning before my stroke. So this shows damage to the right side of the brain affecting the left side of the body. Now, the parietal lobe, you'll recall that was behind the frontal lobe. The parietal lobe on both sides of the brain plays a strong role in spatial awareness, and the right side parietal lobe plays an even bigger role in attention. We rely on our right parietal lobe to help us notice the environment on the left side of our body. Therefore, a right side parietal lobe stroke is likely to result in left side neglect. That is, you tend to ignore or not pay attention to things on the left side. The biggest symptom of left neglect is the difficulty in noticing things on the left side of the body and the environment. So you may only eat things from the right side of the plate. You may read and write only from the right side of the page. And you may ignore caregivers when you're approached on your left side. So this shows, for example, food is only eaten on the right side of the plate and not the left, because basically you don't see or notice the left side of the plate. Here's what happens when the person has left side neglect and they're asked to draw a clock. You may or may not have been asked to draw a clock to assess your stroke. I know that I was asked to draw a clock. I know this is not the one that I drew. Mine was perfect. But this one is obviously has all the numbers and the hands on the right side. They're completely ignoring the left side. Now, the upper diagram here shows the left hemisphere of the brain, and this dark orange area is primary somatosensory cortex. This little area is secondary somatosensory cortex. Primary somatosensory cortex is often called S1, secondary S2. That's illustrated down here in the bottom slide. Now, why am I showing you that? To talk about Gerstmann syndrome that you might get. Gerstmann syndrome results from damage to the left parietal lobe outside of S1 and S2. The main symptom is an inability to distinguish between your fingers. This is called finger agnosia. Agnosia is a medical term that was introduced into medicine by Sigmund Freud. Freud was a very good neurologist before he became a psychiatrist. So if someone asks you to name a finger, you can't do that. If someone asks you to, for example, raise your index finger, you can't do that. You can't point to a particular finger on a chart. If someone tells you to point to a little finger, you can't do that. You also have difficulty in writing and drawing, and you have problems with mathematics. Now, it's very interesting that you have problems distinguishing between your fingers and you have problems with mathematics. You might recall that you probably learned how to count by using your fingers. Artie, did you use your toes? I guess not. But people normally learn to count on their fingers. There seems to be a link between mathematics and your fingers. And that's demonstrated with Gerstmann's syndrome. Okay? Now, have you ever felt like this? You're running on empty. You don't feel like doing anything. This is called post-stroke fatigue. Now, fatigue after a stroke differs from normal tiredness. While some people feel fatigue, feel better after a nap, stroke survivors with fatigue might not feel energized even after a full night's rest or daily naps. Post-stroke fatigue can affect anyone, young or old, active or sedentary, male or female. The exact cause of post-stroke fatigue varies from person to person. One example is the body uses energy differently after a stroke. For example, if a leg has become difficult to move, it takes more energy to move around. If you struggle with post-stroke pain, that requires an enormous amount of energy to cope with. This can deplete your energy, leading to post-stroke fatigue. Fatigue after stroke can also be the result of emotional factors like grief, depression, or other emotional changes. Also, the size of a stroke is not related to the severity of post-stroke fatigue. Someone that survived a massive stroke may have little fatigue, while a mild stroke survivor may feel tired all the time. How do you manage post-stroke fatigue? Well, talk to your physician. Check your medications. They may have side effects of drowsiness. Also, try to conserve your energy by prioritizing what's most important. Plan the best way to do something. Pace yourself by spacing out more taxing activities. And posture. Pay attention to your posture. You might also want to keep an energy journal. You might improve your stamina by practicing something more and more. Eat well and sleep. Sleep is extremely important for your brain to heal after a stroke. Focus on one thing at a time. Multitasking is a myth. You do best when you deal with one thing at a time and try to manage any emotional changes. What about stroke recovery? Some spontaneous recovery does occur after a stroke, but it won't last forever. Spontaneous motor recovery only occurs during the first 6 months of recovery. Afterwards, rehabilitation is necessary to make further progress. There's a remarkable capacity of the adult brain to undergo plasticity that promotes recovery from stroke damage. and there are parallels between plasticity mechanisms in the developing nervous system and those taking place in the adult brain after a stroke. Which brings us to neuroplasticity, the brain's ability to change or learn by forming new neural connections throughout life. It is neuroplasticity that allows neurons in the brain to compensate for injury. Now, I put a question mark up there after stroke recovery because re-emergent post-stroke behavior is unlikely to be identical to pre-stroke behavioral patterns because of the loss of neurons that have highly specific functions. And the commonly used behavioral assessment protocols can rarely determine the extent to which improved performance reflects true recovery, behavioral compensation, or a combination of both. When you go in for therapy, you're often given some assessment tools like putting pegs in holes, and they time you on that. And then when you're done with your therapy, they'll give you the same test. Now, what they're measuring is the length of time it takes you. But that doesn't say whether you're using the mechanisms you would use with your good hand when you're using your bad hand. You may get better with your compensation with your bad hand rather than learning to actually use your hand the way you did before your stroke. What are stroke recovery mechanisms? Well, the motor and sensory cortices, and I showed you those at the outset, the motor cortex in the frontal lobe and the somatosensory cortex in the parietal lobe are loosely organized into what are called somatotopic functional maps. Somatotopic means sort of the body's mapped out on the brain. And they show high levels of use-dependent plasticity. That is, the maps can be modified by experience. The motor maps reflect the coupling of specific motor cortex neurons to specific muscles. The sensory maps represent the pairing of sensory information from particular body parts to specific sensory cortex neurons. And remember, this dark blue up here is where you have a motor map of your body. And the primary somatosensory cortex, where you have a sensory map of your body. And that's shown here. Each map is called a homunculus. And this illustrates a motor homunculus. This is the way your motor cortex sees you. That is, in terms of its ability to control individual body parts. So you normally have a very large amount of cortex developed to your hands, your face, especially your mouth and lips. After a stroke, the body part affected has its homunculus reduced in size, but that size can grow again if you use it. You use that body part. Now, there are two related factors that help plasticity in the adult brain after stroke. First, there's a lot of redundant connectivity in the brain. Second, new connections between neurons can form in the brain throughout the lifespan. In general, body parts are controlled by neurons in the cerebral hemisphere on the opposite side of the body. But you do have ipsilateral pathways in which, for example, the right hemisphere controls the right side of the body. And you can make use of these ipsilateral pathways during recovery to help your improvement. You can also use intact neurons in the opposite hemisphere, which is sort of what I said. But recovery of language in patients with aphasia depends on reorganization of neurons in the left hemisphere. Now, I talked about this study before in an earlier talk I gave, but it makes sense to cover it again. Edward Taub studied patients with strokes that impaired the ability to use one arm and hand. The unaffected arm, that is the good arm, was put in a sling for 14 days, forcing the patients to use the affected arm. This procedure is called constraint-induced movement therapy, or CI therapy. The control group received cognitive relaxation and physical fitness exercises for the same amount of time. It was only the constraint-induced movement therapy group that had long-term improvement. And that's shown here, and I showed you this figure before. You can see the improvements in the orange line there in the group receiving the CI therapy compared to the placebo control. And in general, exercise builds brain health. Kaufman and colleagues in 2007 reviewed research showing that exercise can improve learning and memory, delay age-related cognitive decline, reduce the risk of neurodegeneration, and alleviate depression. And they pointed out that inflammation is inhibited by exercise. Reduction of inflammation by exercise is how exercise reduces the risk factors for cognitive decline and neurodegeneration. That's shown in this slide. Exercise helps to prevent AD, that's Alzheimer's disease, PD is Parkinson's disease. It helps eliminate inflammation. It improves growth factor induction and signaling cascades. And this, which I think I showed you before also in a prior presentation, points out that exercise, like antidepressants, increase norepinephrine and serotonin and BDNF, brain-derived neurotrophic factor. BDNF is like miracle grow for the brain, and that makes your neurons happy and healthy. So in general, instead of thinking use it or lose it, think use it to improve it. That should be your mantra in recovering from stroke. Use it to improve it. And here are some suggested readings. Stronger After Stroke by Peter Levine, now in its 3rd edition. Also the book The Brain That Changes Itself by Norman Doidge. And finally, My Stroke of Insight by Jill Bolte Taylor. And that concludes my presentation for today. I sincerely hope you've enjoyed it. I've enjoyed presenting to you. Thank you very much.
Thanks taking time to having session for a session for all of us. Thank you so much. But I leave it to people to ask their questions if there's any. There are quite a few people out here who are coming to the session today.
I'll be happy to answer any questions you may have.
Yeah, that's what. So as of now, I've seen questions. Yes.
Yeah.
Yes, Unnati.
Yeah. Thank you, sir. Thank you for the wonderful session. I had a few queries. You just spoke about post-stroke fatigue. After 4 years, right now, my husband, he is feeling the same, but he is feeling drowsy. He feels he wants to sleep for some time. This I have observed as a cycle which happens once in a year, maybe 10 to 12 months around, and for a month he is like that. Then again he will come back to normal. So is it post-stroke fatigue or due to change of some routine or weather? I just wanted to know.
Well, post-stroke fatigue affects many stroke survivors, and unfortunately, resting or sleeping doesn't seem to really help it. The best way to help it is to improve whatever the problem is. If it's motor, then work on motor recovery. If it's thinking skills, and do thinking exercises and try to plan your days so that what's most important you get done first. And don't try to multitask, because that really doesn't work even for people that haven't had a stroke.
Yes, I think that's right. And it's very suitable to adopt. So one more thing I wanted to ask was, is ischemic stroke, the occurrence of ischemic stroke is greater compared to the occurrence of hemorrhagic stroke. What I heard was, if there is a left hemisphere stroke, the clot would have probably traveled from the heart. Is it true that way?
I didn't completely understand you. I'm sorry, there was a little static.
I think she asked, in the case of ischemic stroke, the blood clot, they say it travels either from the heart. It's thrown out from the heart. Is it true? That's what she wanted to know. In case of ischemic stroke, does the clot come from the heart?
Oh, a blood clot can come from anywhere, but it often comes from the heart. For example, people will often have atrial fibrillation in their heart. And atrial fibrillation will cause a blood clot to be released, or can cause a blood clot to be released from the heart, and that can get to the brain and cause a stroke. So, but blood clots can be released from anywhere. For example, people may discover that they have a blockage of their carotid artery or another artery, and they could either get that treated via a medicine or surgical treatment. And unfortunately, sometimes if they opt for surgical treatment, during the treatment, something breaks off from the artery and gets into the brain and causes a stroke. I know of someone who had an abdominal hematoma, that is a bulge in an artery associated with their stomach, and they went in for surgery to have that hematoma removed. But in the process, a bit of something broke off and caused a stroke. So the abdominal hematoma was fixed, but now they're living with the consequences of the stroke that was caused by the surgery in their abdomen.
Oh,
Okay.
okay.
Hardeepji, you were saying something?
Yeah, I just wanted to add in this. We do have on our group also someone who had apparently a clot in the leg travel up and through veins or arteries perhaps and cause stroke. Secondly, what you mentioned during a surgical process, actually that's what happened to my mother. While undergoing stenting, unfortunately the clot in the carotid artery broke down and traveled upstream and stroke. So she actually suffered a stroke during a surgical process. While
I'm sorry to hear that, but I've heard that for several people that
yes,
they opt for a surgical improvement, but the surgery causes a stroke because something
but
breaks
the
off.
surgery itself has certain risks and sometimes unfortunate, that happens. I was just curious, Bruce, it's a whole lot of information, that you put forward. So was it part of your, let's say, studies of your profession earlier that you read so much about the brain, or is this something that you collated after your stroke?
I'm sorry, I had difficulty hearing you. Maybe it's my speakers.
What I was asking was, in this presentation, you have put so much of detailed information. So is this something you collated after your stroke, or was it there earlier, as part of your studies and your profession as well?
This is something that I just touched on in a very minor way before my stroke. So I had the knowledge of brain structure and function, but not the relationship to stroke until after I had a stroke. So I put this all together after my stroke. But I did have a lot of these slides of the brain structure from before. I used those for teaching.
Okay. Also, I wanted if you can elaborate a bit about chronic pain. You mentioned about, in the beginning.
Yes, there is post-stroke pain, and initially it was found when the thalamus was damaged, and it was called thalamic pain syndrome. Now, my stroke was actually in my thalamus, and I was very lucky that I did not develop thalamic pain. There is no known drug treatment for it. The only treatment that's proven effective has been mindfulness meditation. And it's not just damage to the thalamus now that can cause it. It doesn't be caused by damage in subcortical areas, that is, areas below the cerebral cortex. And again, unfortunately, there is no known drug treatment for it, but this mindfulness meditation. And I'm just very thankful that I never developed it.
Thank you. And yes, meditation is important. We do emphasize about that even in our groups occasionally. Thank you, Bruce. Anyone else has any questions or queries? They can come in. I'm seeing Abhiramji there. Maybe you can unmute yourself. Kilaji can moderate.
Hello, sir. Good evening.
Yes, sir. Please go ahead.
Yeah. No, it was very innovative, interesting. And though I'm a doctor, I have not really given a thought to so much of brain function since it's not my domain of subject. But it was really, really inspiring as well as very informative. And thanks to sir for that. I think only a survivor can talk more about the stroke, as he has passed through that suffering and recovery, which is quite commendable. Thank you, sir.
You're most welcome.
Indeed.
Professor, this one question. So for me, because I do have a pain in my right leg from the time I had the stroke. Okay? And of course, when I go to my physiotherapy, he's happy to say that this will get better with time. Okay, we don't have any. Okay, I think, is it true, right? Is it true?
Is this the pain you were referring to? Is what he says. Like if he's walking and all, it's fine. If he just sits down for some time and then immediately there is a pain in the legs. So is this the type of pain you are saying? You refer to thalamic pain. Is this the kind of pain you are saying, or which other pain?
I didn't quite understand the question.
No, my husband, he had a stroke. The left parietal lobe was affected. Okay. And so now he experiences weakness on his right hand and right leg. and there is pain when he sits down for some time. There is stiffness and it pains. So is this the type of pain you were referring to? Is what we want to know.
So his stroke affected the left side of his body, but he's got
Yes.
pain on the right-hand side.
Yeah, correct.
Yeah, I don't think that has anything to do. That's not what I would think of as post-stroke
Not
pain.
okay.
No.
Okay.
Now, I don't want to pry here, but for example, people with diabetes will often have peripheral neuropathy, and they may feel pain or numbness in their limbs from the diabetes. But
Okay.
if the stroke affected the left side of his body and he had pain on the right side, that is not post-stroke pain.
That's not okay. Okay. And also, I wanted to ask about emotions. Are they heightened after the stroke? Emotions like display of, are you overly emotional in your reaction? Like a person who used to be, very calm, and suddenly you start crying and you're so scared to face situations and extremely emotional about even small things. So is that
Emotional changes can occur, and they can be drastic changes after a stroke. Emotional changes may occur from damage to parts of the limbic system or the prefrontal cortex
prefrontal cortex?
up here. And people may become, well, you can get almost any change. I know of one person who now is much more calm and pleasant and talkative after a stroke.
Okay.
While another that I heard about is very short-tempered, and almost mean after the stroke. So emotional changes can occur. I'm not sure what can be done to treat that. Most stroke treatments deal with movement problems.
Yes.
Okay.
Thank you.
Mhm.
Yes. Any more questions? Anybody has?
I think Rajinder ji has raised his hand.
Rajit. Yes.
If you can unmute yourself, Rajinder sir,
Yeah.
and speak up if you can. I think he had some issues with his system. You can put in the chat box also, Rajinder ji, if you have any query. Okay, while we wait for him to come back, I had some queries that I was noting down. You talked about, Bruce, I was interested, if you can elaborate a bit. You said something like there is no penumbra in a hemorrhagic stroke.
Hemorrhagic stroke?
There is no penumbra. That's what you said.
Yeah, well, there is no penumbra. That is correct. There have been several studies that have shown that there is no penumbra. And I'm not sure why there is no penumbra, but there isn't.
Okay. Whatever is the damage is kind of a complete infarct. It is those completely die. There are no.
In a hemorrhagic stroke, the core is still damaged. And that's dead. But in a sense, I was somewhat lucky in that regard, because my stroke was in my thalamus, but in the part of the thalamus that was closest to the middle of my brain. And without getting too detailed, along the middle of your brain, you have what are called cerebral ventricles, or spaces that are filled with cerebrospinal fluid. And a lot of the blood from the hemorrhage from mine went into the ventricles. So a CT scan of my brain showed that, let's see, the ventricles along the middle were completely filled with blood. But luckily, they were filled rather than having that blood go out and press on other parts of my brain.
Indeed, that was fortunate, as you say, that other parts were not damaged because of pressure.
Yes.
That is true. Yeah, I've seen. Fine. That was some of the things that I noticed, and of course, a huge amount of information, Bruce, which will be very useful, I'm sure, going forward also. Just another thought which comes to my mind, and maybe others can also come in here. When one suffers a stroke and one is discharged, do the doctors go into such details like what we just saw here, that these are the areas damaged, these are the possible consequences that you might have, the improvements or the deterioration going forward? What is the experience of others also, if they can share? Do they get into, because here I'm seeing so much of, pretty much the whole of brain has been mapped, and each area, even the slightest bit, controls something, affects something. So when we are discharged, Bruce, when you were discharged, for example, or were you told that this is exactly what happened and this is what you can expect going forward?
I didn't quite understand your question.
I was talking about, do the doctors at the time of discharge indicate to the patient that these are the brain areas which are damaged, and so probably this is what may happen in the future, or this is what is happening to you?
Well, in my case, the doctors got it wrong. Well, I mentioned that my primary care physician later told me, when he first saw the CT scan of my brain, he assumed that I had died. So that was clearly wrong. Also, now, I didn't elaborate on this today, but after it was determined I had the hemorrhagic stroke, I was put into a drug-induced coma for 8 days. I was in a coma because of a medicine I was given. And the attending neurologist at that time told my wife that if I recovered, she should prepare herself, because I would not be the same person that I was before I had the stroke, that I would be completely changed. And he was wrong also. I have the same personality that I had before my stroke. Now, I'm glad they put me into a drug-induced coma, because that kept the neurons in my brain quiet and not being active. And after a stroke, for a couple of days, a stroke can get worse because, now this is a little complicated. I'm not sure it will make any sense. But when neurons in the brain are dying or damaged, they will often release their neurotransmitter at the synapse. And one of those neurotransmitters is called glutamate, that excites the cell that it's released on, and that cell becomes active. But too much of the glutamate can kill the cell that it's released on. It's neurotoxic to other neurons. And that can cause a cascade of death emanating from the center of the stroke. That is, neurons at the center, when they're dead or dying, will release glutamate, if they're glutamate neurons, and that will excite and maybe kill another set. And they will release their glutamate and kill others. It's like if you throw a rock into a pond, you get a rippling out from the center. And so for several days after the initial stroke, symptoms can get worse because of this glutamate cascade. And that was prevented in my brain because I was in a coma and my neurons were not active for 8 days. I don't know if that makes any sense, but I'm glad that they did it.
Very interesting. Very interesting. Didn't realize that coma you had was drug induced. When I read about it in the synopsis, I did not realize that the coma was drug induced. And very interesting to note that how it helped you actually. Very interesting. Just want to thank you. Thank you, Doctor.
Thank you so much, Professor, for taking time today. Okay, I don't think there are too much other questions that come in today. But thanks a lot. Thanks, Mr. Loading, too, for getting this.
You are very welcome. It was my pleasure.
Indeed, we are past almost at 8:30 Indian time. And yes, a
11
lot of
o'clock
Yeah,
in,
it's 11 here for us.
Thank you very much, all, and thank you, Bruce, again for taking the time and the efforts. And hopefully, we will learn a lot from here. And of course, thank you very much, everyone, and Bruce.
Thank you.
Thank
Thank you
you,
so
sir.
much.
Thank you.
I'm closing
Thank
the
you.
meeting
Bye-bye.
now. All the best, everyone. Take care. Thank you.
Thank you.
Thank you.
Bye-bye.
Thank you.
Thank you.
Have a nice day, Professor Bruce, and all others.
Bye-bye.
Bye-bye.
Bye-bye.